Today I will be presenting our investigation of the grammar rules of transcription activation domains. Regulating transcription is critically important for proper cellular function. We know that transcription factors play a key role in this regulation, but it still remains unclear what constitutes the activation domains of transcription factors. Activation domains have no specific sequence or structure. In fact, 1 to 3% of random sequences can actually function as activation domains. Here I will describe the grammar rules for activation domains that we have uncovered by simultaneously analyzing the activity of over 10,000 designed prospective activation domain sequences in a yeast reporter strain. Functional AD sequences permitted growth by activating expression of an antibiotic resistance gene. By sequencing the yeast population over time, we could determine the growth slopes for yeast with each sequence. Sequences were considered to be functional activation domains if they produced growth slopes at least 2 standard deviations above that of the average slope for the negative controls. Previous investigations have revealed that known activation domains have a bias for acidic and aromatic residues. In analyzing libraries of random sequences using machine learning models, we observed previously that functional ADs are enriched in acidic amino acid residues D and E and aromatic residues W and F, as well as being depleted of basic residues K and R. In the current library, we observe that simple dipeptide repeats are functional if they consist of aspartic acid D alternating with tryptophan W, phenylalanine F, or leucine L, as seen by positive growth slopes. Additionally, we observe that a functional sequence comprised of D and W residues can be made less and less functional by the addition of more and more basic R residues. This library of sequences was divided into several sets that encompass entire combinatorial spaces with a focus on hydrophobic and acidic residues. For example, the intermixed template is 10 residues long, where every other residue is D. The remaining 5 residues are all combinations of the hydrophobic amino acids W, F, Y, or L. This results in a total of 1,024 sequences in each set. The flank template has D residues located on either side of the hydrophobic residues, and the amphipathic helix template spaces out the acidic and hydrophobic residues with glycine. Additionally, we analyze sets comprised of individual hydrophobics paired with D across 10 positions. We refer to these sets as WD10, FD10, LD10, and YD10. One key result, which is consistent with the known diversity of AD sequences, is that a large portion of the sequences in the flanked, intermixed WD10 and FD10 sets are functional without any identifiable motifs. In general, the most functional sequences in the flanked and intermixed template are enriched in W and F residues, while non-functional sequences are enriched in Y residues. In addition to the composition bias of acidic and hydrophobic residues, we have identified additional composition criteria enriched in functional sequences that we refer to as grammar rules. First, functional AD sequences have balanced amounts of hydrophobic and acidic residues. Secondly, functional AD sequences tend to have hydrophobic residues at the C-terminus and acidic residues located internally. Finally, functional AD sequences have few homoamino acid clusters. In other words, the acidic and hydrophobic residues are more intermixed in functional ADs. We investigated the amphipathic helix template further, and while combinations of W, Y, F, and L sometimes produce slightly functional ADs, the incorporation of proline into this type of sequence produces a very functional activation domain. This result suggests that helical structure is not important for AD activity, as proline is a known structure-disrupting amino acid. While specific sequence motifs are rare across the activation domain sequences we analyzed, in sequences comprised of L and D residues, the most functional sequences contain a specific functional motif with the core sequence L, D, D, L, L. Sequences with this motif are some of the most active ADs tested throughout the entire library. especially when those sequences also followed the identified grammar rules. An alternate approach to understanding ADs is the development of models trained on natural sequences that can predict functional ADs. The Paddle machine learning model is trained on all yeast transcription factors, while the mechanistic predictor model is empirically derived and defines functional ADs based on the net charge and number of hydrophobic residues. Our library's sequences were already biased towards compositions with the potential to be functional ADs, and correspondingly, both prediction models over-predicted functionality. We suspect that these models fail to recognize nuanced grammar rules. This is likely because of the limited number of natural ADs that exist to be used to train the models. We believe that the high diversity of functional activation domain sequences may oppose a traditional recruitment model that describes the role of an activation domain as a specific site of interaction between one transcription factor and another transcriptional regulator or the core transcription machinery. Alternatively, we propose that the activation domains may function as surfactants in a nonspecific fashion, whereby the acidic and hydrophobic nature of the AD can act upon the histone-DNA complex influence gene expression through histone modification. Further investigation into what defines an activation domain and directed biochemical tests are still needed to determine how these grammar rules correspond to biological function.